Okay, today we're going to look at solving dihybrid problems. Many students need a little extra guidance with this. Now, we already have, or you could review for yourself, a good animation that breaks down Mendel's experiments and gives you some straightforward examples. But let's do a quick review if you have saw that video a while ago. So, the parent's genotype, whether it's a parent plant or a plant parent animal, we're assuming it's a diploid organism, so it has two copies of a gene, so it has two alleles for its given gene because it has homologous pairs of chromosomes. During meiosis, the homologous pairs separate, and so in the gametes, you can only get one copy of each gene, so you're forming haploid gametes. Now, obviously, if the individual was heterozygous, you could make two different types of gametes, whereas if the individual is homozygous recessive, you would only be able to make one type of gamete that we still need to show, the separation of the homologous pairs and the separation of the two pairs of the gene. Now, independent assortment. We are going to assume that the genes are located on different chromosomes. When we get to chapter 15, we will talk about when genes are located on the same chromosome. And again, we're assuming the parental genotypes are of organisms that are diploid, like humans, and many plants and many other animals. But now we're looking at two genes. So again, the A's have to separate by segregation, the B's have to separate by segregation, but that means we can get all sorts of different combinations if the parent is heterozygous for both genes. So we're going to break out the Foyle's method that you learned in math class. So 1A, 1B, both uppercase. Uppercase A, the dominant one, lowercase b, the recessive one. Now we could go the recessive A, the dominant B, or we could do the recessive A, the recessive B. So again, we have shown the possible gametes. They are haploid. You only have one A and one B in each cell that is formed. Now, not always is an individual going to be heterozygous for both genes. So let's look at if we have heterozygous for one gene and homozygous recessive for another gene. So we could pass on big A, little b, or big A, little b again. That's a repeat, so we usually only write it once, though it is important to write it twice if you're actually doing a dihybrid cross, because you need 4 gametes to fill up the side of your dihybrid. We could do little a, little b, and little a, little b. And again, that's a repeat. So normally you wouldn't, if you're just listing, you wouldn't list it right there. And again, these are the possible gametes, 1A, 1B, so they're haploid. Okay. Now, rather than using some obscure non-existent gene that we're using A's and B's to represent, or going back to Mendel's pea plants, which I'm sure we are all ready to leave behind and do something a little bit more engaging, let's look at human genetic disorders in this example. So we're going to look at Huntington's disease and cystic fibrosis. Huntington's disease is a dominant allele mutation which begins showing initial symptoms during midlife. It's lethal, and the only reason why it continues in the human population is it doesn't kill people until they've had an opportunity to reproduce and therefore an opportunity to possibly pass on the gene. It is located on autosome 4. We are not looking at sex-linked genetic disorders until we get to chapter 15. The HTT gene provides instructions for making a protein called huntingtin. Although the function of the protein is unknown, it appears to play an important role in nerve cells in the brain. And Huntington's disease is a progressive brain disorder that causes uncontrollable movements, emotional problems, loss of thinking ability, and ultimate death. Cystic fibrosis is the opposite of Huntington's disease in that it is a recessive allele mutation, but it is still lethal. And it usually kills in childhood, though with modern treatments, some people can live into early or mid-adulthood. Where is it located? On a totally different autosome, autosome 7. The technical name for it is the CFTR gene, and it provides instructions for making chloride ion channels. Mutations in the CFTR gene prevent the channels from regulating the flow of chloride ions and water across the cell membranes. As a result, cells that line the passageways of the lungs, pancreas, and other organs produce mucus that is unusually thick and sticky. The most common symptoms include progressive damage to the respiratory systems and chronic digestive system problems. So here we are at our dihybrid human genetics problem. What are the possible outcomes if the following two 19-year-olds find out that they are expecting a baby? Sally is heterozygous for Huntington's disease and a carrier for cystic fibrosis. John is homozygous recessive for the HTT gene, but he does have cystic fibrosis. So what alleles shall we use? First of all, we want to make sure we use alleles that we can actually tell the difference between capital letters and lowercase letters. So Huntington's disease, that's pretty easy. Capital H for Huntington's and lowercase h for no Huntington's. Cystic fibrosis is a little bit more complicated because if we wanted to do cystic fibrosis, that's C, or if we wanted to do chloride ion channel, that's C, and C is hard to tell, and also usually use the letter of the dominant. So let's go with F. F for functional chloride ion channel preventing the development of cystic fibrosis. So we're just going to call this no cystic fibrosis. Though you do have to be careful with F's. If you're not careful, F's can look alike. And we're going to use lowercase f for cystic fibrosis. Now remember, these are the alleles. Okay, you have two options for each gene in this case, and these are again alleles, alleles. So now that we've got our letters that we're going to use to represent the alleles, let's look at Sally's genotype. So Sally is heterozygous for Huntington's disease. So hetero means different. So she has big H, little h. Probably at 19, she's not showing any symptoms of Huntington's disease, but she will develop it in her future. And she is a carrier for cystic fibrosis. So she does not suffer from cystic fibrosis because it is a recessive disorder. And so she has one good copy of the chloride ion channel information, so she can make functional chloride ion channels for her mucus-producing organs like her lungs and her pancreas. But she does have hidden in her genetics one recessive copy of the cystic fibrosis genetics, which means if we follow the FOIL method, she could pass on. I'm going to just change colors. Big H, big F, or big H, little f, or little h, big F, or little h, little f. I'm a little worried that that light blue is not showing up, so I'm going to change the color back to red. So I'm going to put commas in between each of these to indicate that they are separate from each other. Notice the eggs are haploid. I'll even put them into cells, haploid cells. Now John's genotype, let's look at John. John is homozygous recessive for the HTT gene, which means he does not have Huntington's disease. So he is little h, little h. But that doesn't really matter because he has health issues right here and now because he does have cystic fibrosis, and the only way you get cystic fibrosis, which is a recessive disorder, is if you have two little f's. So here we go. He has the ability to make one type of sperm. That's all he can make. So what are the chances? Let's take a look here. What are the possible probabilities of the offspring's phenotype? Go away. So I normally put the female up here, so this is going to be Sally's eggs. Okay, inside the Punnett square are the possible offspring. On the side, those are going to be John's sperm. And so Sally's eggs can have big H, big F, big H, little f, big F, and little h, little f. John's sperm, little h, little f, little h, little f, little h, little f, little h, little f. That's all he's got. So we're going to be figuring out all of the possibles for this child. So I usually will go through, oops, make that go away, and fill it through. So if this egg is fertilized by that sperm, big H from Sally's egg, little h from John's sperm. Big F from Sally's egg, little f from John's sperm. Okay, if this egg is fertilized by that same sperm, it's going to be big H from Sally's egg, little h from John's sperm, little f from Sally's egg, little f from John's sperm. If it's this egg that's fertilized by that sperm, then we can say it would be little h from Sally's egg, little h from John's sperm, big F from Sally's egg, little f from John's sperm. If it's this egg, on the other hand, then it's going to be little h from Sally's egg, little h from John's sperm, little f from Sally's egg, little f from John's sperm. Now, if we had more complicated genetics for John's possible sperm, we would have much more to fill out in this Punnett square, but because he can only form one type of sperm, he therefore, all of the dihybrid cross would be identical all the way down, so we don't have to fill it all the way up. Yay! So now, how do we read and turn genotypes? You need to turn genotypes to phenotypes. So we've got genotypes in the dihybrid cross. We want to know phenotypes for the possible baby. So this is where I like to grab my highlighters. And I'm going to say if it's Huntington's disease, it's going to get a yellow slash. So to have Huntington's disease, you need one big H or two big H's. Huntington's, the rest of them not. And again, all the way down, all the way down. Okay. And then change the color. Let's say green for no Huntington's. No Huntington's. The only way to get no Huntington's is two little h's. So two little h's, no Huntington's. Okay. And then let's see. Let's go ahead and pull some blue. That'll work. No cystic fibrosis, no cystic fibrosis. The only way you get no cystic fibrosis is if you have one big F or two big F's. That's one big F. That's two little F's, skipping it. That's one big F. And that's enough. And then we need one more color. Let's go with light gray. Cystic fibrosis, cystic fibrosis. The only way you get cystic fibrosis is if you have two little F's, two little F's, two little F's. And oh, and again, it's all the way down. So, grabbing our pen again, we see that how many babies out of 16, how many possibilities out of 16 are there that you'd get a baby with Huntington's disease and no cystic fibrosis? 1, 2, 3, 4 out of 16, which of course I know you could reduce down to 1/4. Okay, or 25%. What are the chances of getting a child with Huntington's disease and cystic fibrosis? The double whammy. So that's a yellow and a gray streak. 1, 2, 3, 4, 4/16. What are the chances of having a perfectly healthy baby with no Huntington's disease and no cystic fibrosis? So the green and the blue. 1, 2, 3, 4, 4/16. What are the chances of getting no Huntington's disease but cystic fibrosis? 1, 2, 3, 4, 4/16. Or from another perspective, you have 12/16 of a chance of having a child with one or more lethal genetic conditions and only a 4 in 16 chance of getting a child with no lethal genetic disorders. Now I would like you to try it for yourself again, but now let's make it a little more interesting. Let's say that John is homozygous recessive for the H2T gene, so again he's healthy, but this time he does not have cystic fibrosis. He's a carrier for cystic fibrosis. So at this point, both Sally and John think they are perfectly healthy unless they know their genetic history. Go ahead, give it a try, see if you can figure it out.